So like we said earlier on, we have a difference in terms of what happens in plants and also in animal cells. And we have explained that thoroughly. But we can basically review. The first thing is to know the pressures that will happen. We earlier on talked about the pressure potential, which we said is the pressure that is exerted by the cell wall or cell membrane, and that will basically prevent movement of further particles into the cell. We talked about that earlier on. Now here, we are saying that target pressure is the force exerted on the cell wall by the cytoplasm due to the water entering the vacuole, causing it to expand. And this pressure helps in maintaining the shape and the rigidity of the cell. And this is particularly very important given the fact that plant cells actually have the cell wall, and that cell wall is very important in shape, maintaining shape, and also that rigidity. And now when you have a hypotonic solution, it has less solute concentration, okay, than the cell sap. And here we are talking about some parts of the cell. And we are talking about, for example, the cell sap obviously can be vacuole, cell vacuole. It is in the cell vacuole that we are having the high solute concentration. That is where we are having high solute concentration. So we are relating the surrounding solution and what happens or what is in the cell vacuole. Now, when you have a hypotonic solution, it means that the water is basically entering the cell by osmosis, and therefore the cell will basically swell. Okay, so you will have the cells becoming turgid, and this is very important in maintaining support. So when the water enters into the cell vacuole will expand, and when the cell vacuole basically expands, it will push against the cytoplasm, and that same pressure is transmitted to the cell wall. Okay, so that is the target pressure that we are talking about. So when the cell is fully turgid, we say that it has full target pressure. Okay, so it has taken in a lot of water. Okay, so that's what you will see. The sap vacuole will increase in size because it has taken in more water, and then that vacuole will exert a pressure on the cytoplasm, and then that pressure is the same pressure that you find on the cell wall. Okay, so that is turgid pressure. And then when we talk about the sap vacuole, why do we refer to it? Plant cells actually have large sap vacuoles. Animal cells may actually lack or even have very small vacuoles. So what happens is that in a plant cell, you have the sap vacuole being placed centrally or in the center. It has a lot of solutes. So these are basically salts and organic compounds like sugars. And that is what lowers the water potential. Okay? It lowers the water potential. That means that if you have a lower water potential, these water molecules in the sap vacuole don't move out. Okay? So these salts and organic compounds make the water potential more negative. And that is basically the tendency of water molecules moving becomes very limited. And that's why the water has to come in to the cell. Okay, so that's why the sap vacuoles are very important. They contribute a lot to the turgidity of the cells. And remember, turgidity of the cell is very important in maintaining support in herbaceous plants. Okay, so that is what we have there. Cell wall pressure. You can call it the wall pressure. It refers to the force exerted by the cell wall on the cell contents in a plant. So remember, we are having, if you have a cell that is placed in a hypotonic solution, it takes in that water. So this sub-virtue expands. When it expands, we are saying that it exerts pressure on the cytoplasm. Okay, and that pressure is again transmitted to the cell wall. But the cell wall will also counter that pressure. Okay, because remember, action and reaction are equal, but what? Opposite. So you have another pressure, and that pressure is now the wall pressure. So remember, the cytoplasm is pushing, but also the wall pressure is also what? Pushing in the opposite direction. Okay, so that's why you are having here target pressure or target pressure in that direction, and then the wall pressure also in the opposite direction. But you also have osmotic pressure that we are also having in that direction. Okay, so that is what basically happens. So these are basically opposing forces: target pressure, cytoplasm content, and then wall pressure, the wall trying to exert pressure. Okay, on the contents of the cell, basically the cytoplasm and its contents. So the osmotic pressure, which we saw there, is that it's the minimum pressure that is needed to be applied to a higher concentration solution to prevent movement of water into it from the lower concentration solution across the same chemical membrane. Water molecules will not move endlessly into the cell. Okay, so there should be a point where you have a stop. And we are basically saying that osmotic pressure puts a stop to this. So osmotic pressure prevents the movement of water into a particular solution, especially if you have this movement happening from a region of lower concentration solution. Okay, so plasmolysis is what happens when you have a plant cell being placed in a hypertonic solution. It will lose its water content into the hypertonic solution because of the difference in water potential. When you have a hypertonic solution, it means that the water potential is negative. So the tendency of these water particles or molecules to move is actually less. And therefore, instead, the water molecules will move from the cell into the hypertonic solution. And therefore, you will have shrinkage where you will have you will have a plasmolized cell. So with the plasmolized cell, you have the cytoplasm losing contact with the cell wall. That means, therefore, that the target pressure is actually reduced. Okay, target pressure is actually reduced because that target pressure comes when the cytoplasm exerts a force on the cell wall. Okay, so the cell becomes plasmolized, and the key thing is that the cytoplasm loses that contact with the cell wall. So the cell can be called plasmolized cell or flaccid cell. Okay, so that is what you can have there. That in a normal cell where you don't have net movement of particles, that is in isotonic solution, you have that normal cell with its normal shape. In a hypertonic solution, you will have the water moving from the cell into the hypertonic solution. So you have osmolysis. So you have like we have always saying, when you have target cells, it means that they are going to maintain their shape. And some plants, like for example, the herbaceous plants, rely on cell turbidity to basically remain upright.